All right, let's resume our symposium. And hopefully everybody can get back to your seat. The back is not on. I think it's on. Hello, everyone. We're getting back to the symposium. And our next speaker is Dr. Bill Noble from University of Washington. He will talk about how he uses foundation models for chromatin 3D architecture and proteomics mass spectrometry. Thank you.
Thank you. All right, so I'm going to get more into the nitty-gritty rather than sort of the, bird's eye view, and just tell you in detail a couple stories. One of the challenges here is that, I couldn't decide whether to talk about genomics or proteomics, so I tried to talk about both, and it's sort of a 50-minute talk that I squashed down to 25. And then I got here and they said, actually, you should talk for 20. So I might be doing some cutting as we go, just to see how it goes. But I'll start with the genomic side. There's a common thread between both stories, but that'll become clear later. And just by way of motivation, I hope this is not a surprise to anyone, but when we think about the genome sequence, it turns out that it's not just the sequence that matters, but the three-dimensional configuration of the DNA in the chromosome actually has, or in the nucleus actually has important functional consequences. And then it's relevant for fundamental processes like obviously gene expression, DNA replication, and so on. So this is a model of the genome architecture of the Plasmodium falciparum parasite. And there are many ways to look at chromatin architecture, 3D chromatin structure. One of the most popular ones, certainly from the sequencing side of things, as opposed to the microscopy side, is the Hi-C assay. And I'm sure many people are familiar with it, but let me just make sure we're all on the same page. Hi-C is a method that works by using paired-end sequencing to count pairs of proximal DNA loci. The idea is you introduce some crosslinks, cut with a restriction enzyme, and then you're going to fill in the ends of the cuts with biotin, and that allows you to then pull down these sort of molecules here that kind of look like doggy chew toys. And the key thing is that if you've ligated two loci that were far apart along the DNA sequence, but close in 3 dimensions, you can map those back to the genome and figure out which things were close in 3D in the cell. So you do this for a population of cells, and the primary data object, it's usually represented as a matrix like this. So this is a Hi-C contact matrix. Just like any other contact matrix, the values indicate proximity of two different entities on the axes. In this case, they're fixed width genomic bins. This one particularly is the yeast genome. And what you can see is, high values mean there were many pairs of sequencing reads that connected locus I to locus J. Okay, so that's the kind of data that we're interested in. One of the things that became clear, so this assay was first described in 2009. It wasn't until about 2012 that people figured out that in addition to seeing sort of chromosome territories and large-scale activating and repressive compartments, with enough sequencing, you could also see things like domains, like gene size domains that maybe are relevant for gene expression. And then a few years later, as you got even more sequencing, you could start to see punctate phenomena like loops that might correspond to sometimes enhancer-promoter interactions, these kind of things. So there's all these different scales that are all visible in principle from the same initial data matrix. And as a result of sort of the organic growth of this field and lots of different people doing the analysis, when you're asking all these different questions, different people have developed different tools to answer them. And so if you're asking a question about domains versus loops versus compartments, you might answer them with completely different pieces of software. And they might have fundamentally different approaches to this kind of data. So like these are just 3 examples, somewhat random examples, but one of them is if you're measuring similarity between pairs of experiments, which is an important problem in this area. Some people will use like a random walk kind of approach. Or if you're going to do resolution enhancement, you might use a deep neural network. Sometimes if you're looking for loops, there's one of the most popular algorithms called hiccups, is sort of a bespoke algorithm specifically developed for analyzing this kind of data. and there's nothing wrong with any of these individual solutions, but you have the feeling that by treating each phenomenon independently, you're kind of leaving something on the table because it's all the same data under the hood, right? So the idea of our project was that we'd like to sort of follow this foundation modeling approach that's been successful in other scenarios. So we've talked about this already, but the idea of a foundation model, the canonical example being these large language models, is that you're trained on 10 trillion different tokens, different strings of natural language. Typically in a self-supervised way with maybe that kind of masked language modeling approach that we talked about earlier. And the idea is you train on a huge amount of data and then you can apply it to lots of different downstream tasks. And because all this prior knowledge has been ingested into the model, it can be quickly fine-tuned with relatively cheap compute to do things like writing poetry, answering multiple choice questions, summarizing documents without too much overhead in terms of the data requirements or the computational requirements. Okay, so Xiao Wang was a postdoc who was jointly advised by myself and Sheng Wang in computer science. And this was a collaboration with people from both of our labs, as well as with Sergei Dulatov, whose contribution we'll see a little bit later. So Xiao developed a software system, basically a machine learning system called HiC Foundation. And it follows exactly this sort of foundation modeling approach. So you train it in 2 stages. The fundamental unit is a submatrix of this huge HiC matrix. So we're working with HiC matrices that are typically at 10 kilobase resolution. So 3 billion base pairs divided by 10,000 is the size of each dimension of this matrix. And then the submatrices are each 2.24 megabases in size. And in the stage 1, you're going to just learn just from that data. And then in stage 2, you're going to adapt it to various tasks. So the learning from just that data, we didn't actually invent new modeling techniques. We just said, listen, we know what works. If we pretend that this matrix is an image, then we can just pull existing tools from the literature. So we use this particular architecture known as a vision transformer. And it's, developed for image analysis, but works well here as well. And the idea is kind of like the masked language modeling, except now you're masking out the image. So you're actually masking many of the submatrices, which is what's shown on the left. You put those through an encoder, and then the task of the decoder is to reconstruct the full matrix. One important point in this picture is that the encoder is a lot bigger than the decoder. There's many more parameters in the encoder. It makes this latent representation. The decoder part we don't actually care about. We're not interested actually in reconstructing the matrix. So later we're going to toss out the decoder and put on different kinds of decoders for different tasks. Okay, so imagine we've trained that whole thing. We actually train it on basically all of the available HiC data, which is, it's not actually that much compared to like all the available RNA-seq data. But if you have 1,000 HiC experiments and each of those has, there's a lot of these little matrices, right? So you still have many hundreds of, or at least a couple 100 million training examples to work from. Then we went through and basically showed how this could be applied to a variety of different tasks. And what I'm going to do is I sort of dropped out a lot of the validation slides showing that it works well on these individual tasks. I'll show you what the tasks are, and then I'll just show you one vignette about how we used it in practice. So first task is reproducibility analysis. I think I mentioned this before. This is the task of just saying, did these 2 biologically replicate experiments, did they turn out well enough that they're similar to each other? And then that's an important task, partly just for QC, but then also if you're asking questions about relationships about, different experiments at different developmental time points or relationships among different cell types, you will also need this kind of pairwise comparison. And we train the prediction head for this one using a contrastive learning procedure. So that's what's supposed to be illustrated here in this hypersphere over here. The concept is you put in a bunch of pairs of submatrices from replicate experiments, and then other pairs from randomly paired experiments that are not replicates, right? And the idea is in the latent space, you want the replicates to end up close and the non-replicates to be far apart. And so there's sort of a push-pull component to this contrastive learning where it's pushing the different ones apart and the similar ones together. One way you can, if you look at the literature, there's 4 or 5 different existing techniques that have been developed for doing this. And the way they typically get evaluated is just to say, take a bunch of non-replicates, which are the blue dots on this picture, and a bunch of replicate experiments, which are the red dots, and sort them according to the score assigned by the model. And what you can see here is that the replicates are always well separated from the non-replicates in these different cell types. The performance measure people use is basically the margin, the biological replicate non-replicate margin. And if you take that and compare Hi-C Foundation's approach to some of the existing techniques, these are Hi-C Rep, Genome Disco, and Hi-C Spectre are the names of some of the existing methods. You can see it's performing much better. Task number 2, I'm going to skip over the validation for the other tasks, but I wanted to show you one of them. Task 2 is loop detection, which is arguably one of the ones that's of the most interest to many downstream users of Hi-C. They really care where the loops are, but they're also hard to find because they're sort of a fine resolution thing, so they tend to require high sequencing depth. For that, we used the sort of a trick where we would say, we're going to take a really high depth assay, use an existing sort of robust loop caller to get a set of sort of quasi gold standards. And then we're going to make downsampled versions of this same measurement from 2 different biological replicates that are mixed together at different levels, but with much lower sequencing depth. And then train the model to recover those ones that were called from the high confidence, from the high coverage data set. So we train that prediction head, and then when we want to use it in practice at inference time, there's actually a little clustering step. We use an existing clustering algorithm called mean shift cluster to just make the loop calls. That turns out to work very well, much better than most existing loop callers. Similarly, you can do resolution enhancement just the same way, like, if in a spy movie, you're like zooming in on the license plate and you click enhance, and you can finally read it. We'd like to be able to enhance it and find out where the loops are or the domain boundaries and so on. And so that, again, you do a similar kind of trick. You pretend to downsample, and then you try to get back to the original full data set. We actually also trained this thing to do epigenomic signal prediction, which is not something that had previously been done from Hi-C before, but just the question of, could we use the Hi-C matrix to try to predict what would the CTCF ChIP-seq signal look like, or what would these various histone modifications or local chromatin accessibility profiles look like? It can do that quite well. And then finally, Xiao wanted to do this. I told him it was kind of silly, but he was welcome to try. And then, of course, he showed that I was wrong. It worked well. We'd use this for single cell Hi-C analysis. And the main challenge there is that single cell Hi-C is so sparse that you can't work at 10 kb resolution. Typically, people analyze single cell Hi-C data at like 500 kb resolution or even 1 megabase resolution. So really global scale pictures of full chromosomes. And Xiao said, well, let's just take the full chromosome at that scale and use the pre-trained encoder from bulk Hi-C at 10 kb, and it would still learn to generalize pretty well. So we show in the paper that you can train a resolution enhancement procedure for single cell data, even though the pre-training was done in bulk. So one of the motivations for doing all this was the collaboration with Sergey. Sergey Dulatov's lab is particularly interested in hematopoiesis. And so one of the things that they're interested in is the role of lamin B1, which I'm sure many people are familiar with. It's a very important nuclear envelope protein. This is just a picture on the right showing where the lamin B1 exists along the periphery of the nucleus. It kind of binds some of the DNA to the nuclear envelope. And there's a kind of experiment you can do, a knockdown experiment, where you can knock down the lamin B1 and then check what happens when you differentiate blood cells into neutrophils, right? And there's a procedure here. What's shown here is the neutrophil differentiation, and what you can see on the right is the differentiation of the nucleus into this sort of lobular structure. So that they take on this really weird thing where the chromosomes, some of the chromosomes go into one lobe, some into another lobe, and so on. And it turns out if you do this knockdown, what happens is that if you have the lamin B1 knocked down, then you don't have that. In fact, this corresponds to something called Pelger-Hewitt nuclear anomaly. So it does happen in people as well. So the question is, could we look at data that was generated from this kind of knockdown experiment? And the problem is that there does exist data like this, but it's really hard to get these cells. There's very low cell counts, and so you can't get very much coverage. So the published data was quite noisy. And you can see here, there's one circle, there's one square there that's one potential loop, but it was hard to get high confidence there and hard to see many of them. And so here's what happened when we went back and revisited the data. The raw data is on the left and the enhanced data is on the right. Many more loop calls. Of course, you don't know for sure that these loops are valid, but they're hypotheses that you can use to then investigate further. So we did a series of things in the paper where we looked at, for example, one of the claims in the original published paper was that neutrophils have fewer chromatin loops than the progenitor cells, right? They couldn't get statistical significance for that, but we get a much stronger signal when we look at it from the enhanced data. So that seems like at least additional evidence that maybe there's some validity to that claim. And we can also see, we looked at particular loci that were known to be involved in neutrophil differentiation. And we're able to see particular enhancer promoter loops that were changing differential between the knockdown scenario and the regular control scenario. And then finally, you can do things like gene ontology analysis to say, okay, it looks like all these loops that are gained in neutrophils are actually associated with secretion, which makes sense in the context of this analysis. So they're doing lots of actually other low input high C experiments. So they continue to use this tool in their lab for sort of helping out to analyze high C data at all of these different levels jointly. Okay, so our hope is that High C Foundation will be sort of like a Swiss Army knife for high C analysis, that we're providing sort of a framework. There's a pre-trained model that you can sort of connect to any kind of downstream task that you want. We provide a set of tasks here, the prediction heads that we've trained, but it's relatively straightforward to train a new one. The training of the model itself took, cloud resources and weeks of GPU time, but training each of these individual heads is actually quite cheap. So that's sort of the paradigm that we're operating under. And now I want to switch gears and tell you the other story, or at least part of it. It has to do with a different kind of data entirely. So this is work that was done primarily by Justin Sanders, who's a graduate student in my lab, and jointly with Meli Yilmaz, a former graduate student. And then collaborations with Suwung Oh in computer science, Wout Bitremier, who is at the University of Antwerp, and Nick Riley, who's in chemistry at the UW. Okay, so I'm just going to tell you briefly the outline of this story because there's not that much time left. But the basic idea is that we started by training a de novo sequencing model, right? So if you have a mass spectrum, you're measuring you're basically measuring amino acid sequences, and most mass spectra are identified by searching against a database, but in some situations, you don't have a good database to search against, and that's where de novo sequencing comes in. You're supposed to take the observed mass spectrum on the left and predict the generating peptide on the right. And that's useful in things like immunopeptidomics, but also in things like metaproteomics, antibody sequencing, peptidomics, proteogenomics, paleoproteomics, and phenomics. Yeah, so we treat this as a translation task. I'm going to skip over that. The architecture, it works great. But the main thing that I want to point out is that similar to the high C setting, there's lots of questions that people ask. So all of these, there are papers about, like, what is the generating peptide? That's de novo sequencing. But also, is this spectrum going to be identified in database search? That's what's known as quality control or quality prediction. Does this spectrum contain a post-translational modification? Or is it chimeric? In other words, was it generated by more than one peptide? Or is it from a cross-linked peptide in the setting where you're interested in cross-linking? And so I'm going to skip the example here and go to the main hypothesis, which is our thought, actually Justin's thought was, hey, we've already got this encoder that we've trained for de novo sequencing. Unlike large language models, it was actually trained in a supervised way. rather than a self-supervised way. But in the mass spec setting, that's not that unusual because it's not that hard to get labeled data for mass spec. You can use database search with rigorous statistical false discovery rate controls to get high quality training data. So if, with a big repository of data, you can get about 50% of it labeled relatively inexpensively, just using computational searching tools. And so our hypothesis was we can basically throw out the decoding part of Casanovo and then use the transformer with a new prediction head for a bunch of downstream tasks. And I'm not going to tell you all about all of them, but the basic idea is the tasks that we looked at are spectrum quality prediction, and this green line is the one that's from the foundation model, chimericity, and then we also looked at PTM prediction, and in particular, we showed that if the benefit for the foundation model is most important when you're limited in the amount of training data for the downstream task. So we took a task where there was tons of available data, which is phosphorylation prediction. We get 20 million training examples. In that setting, the foundation model offers no benefit. But if you downsample the number of examples down to a couple thousand or less, which is often the case for some rare PTMs that people don't have much data for, in that case, there's huge benefit for the foundation model. And then finally, the vignette for this one is Nick collaborating with Nick Riley for glycosylation, where they're interested in particular types of glycosylation. And again, the foundation model helps a lot because you don't have many examples of those particular kinds of PTMs. So in both cases, you have a foundation model, a foundation model that you leverage a lot of data in advance and then fine tune it for lots of different downstream tasks. And hopefully it's a general tool that can be repurposed as needed for new tasks as they arise. And I have to acknowledge all the people in my lab who did the work and NSF and NIH for funding, and I'm happy to take questions.
Great talk. I start with one question. It's great to see the Hi-C foundation model. We know the Hi-C still have kind of limited resolution. I see the advantage of the foundation model is to improve the resolution. You did validate by downsampling the reads, but that's, I guess, a slightly different situation, right? You start from even lower resolution to kind of improve the current resolution. There are some higher resolution technique like micro-C or target capture. Did you try to evaluate, from the current Hi-C resolution using those gold standard, how well they can, I guess, impute the loops? And then related question is, how do you quantify the uncertainty? Maybe some estimated loop I have higher confidence, some have lower confidence. Like how is that possible using the external, higher, more expensive data to train such uncertainty quantification?
Yes, good question. I think that's interesting. Well, one interesting question is, micro-C gives you sort of different picture. It's a more proximal to the diagonal, but fundamentally both micro-C and Hi-C are sequencing based, and so they both achieve similar resolutions depending on how much money you have to spend. So the best resolution you can get these days is from DNA Hi-C or some kind of Hi-C that's not bound by restriction enzymes, right? And then sequencing a ton, right? So like the canonical example is Erez Lieberman-Aden's 42 billion reads, right? To get, then he went down to like 10 base pair resolution, some crazy resolution, right? So we tried to use basically some of the highest resolution Hi-C for our analysis. You could, I think, do the same thing with micro-C, but I don't think conceptually that would be that different than using really deeply sequenced Hi-C. I think what's maybe more interesting is like tying it back to there are other kinds of assays, like microscopy-based assays that have fluorescence that give you readouts that I think I would love to try to validate against some of those. It's just a little trickier to do. In terms of the statistical confidence for the loop calling, I think that's a real challenge. Partly, I mean, there are nominal, false discovery rate control built into some of these tools. I think they're very conservative, which is probably the smart thing to do. Like they make these statistical tests, and then if you look at the data, they seem empirically to be quite conservative. we didn't make any attempt to do that for our model because it's not obvious what the right null is. But I think that's a super important question. I think there is some interesting work on, for instance, using knockoffs or something like that to try to get that kind of control, but we haven't made that effort yet. Somebody should do that. Yeah, question.
Hi, Bill. Really interesting work. I have two questions about the high C work.
Okay.
And the first one is, I'm curious about what you mentioned about the decoder. It's a lot smaller than the encoder.
Yes.
Usually when you have a vision encoder, you have a UNet, so the downsampling, upsampling is symmetric. So I'm curious about why it's different in this case.
Right. So in this case, so we're not using the UNet style architecture. So the vision transformer, there's transformers at the beginning and the end, but there's nothing connecting between so the latent representation in the center, there's no connections that bypass that. And I think, you could design it to be symmetric, but we made the choice to basically put fewer layers after because we knew that we weren't interested in the decoding task per se. And that's not something that's specific to the high C setup. People have done the similar things in the image processing field as well.
Okay, thank you. And my second question is there any heterogeneity in high C data between samples, between different conditions, or are you?
Oh yeah, sure, tons of it. But you have heterogeneity in terms of what's the sample, what's the biosample that you're analyzing, but also lots of heterogeneity in terms of the assay itself. It's actually interesting because we're harvesting data from the public consortia ENCODE and 4DN. And so there are variants that go back to, of the assay from 2012. And, so we have all of these different sort of styles of high C that are all being lumped together into this one model.
So does the model use any guidance from those differences?
We don't supervise, we don't tell it, okay, this is in situ high C and this one is intact high C. And, it just gets the data and has to figure it out on its own. On the other hand, in other scenarios, like in the mass spectrometry setting, my empirical observation is that these models don't benefit much from that kind of supervision because it's so obvious what the differences are. They're like, you tell it, oh, look, this is a different type of experiment than that. And they're like, yeah, duh, we knew that. That's obvious.
Okay, thank you.
There's one right behind you.
I was just saying,
Oh, that's nuts.
So hi, Bill. I have one question following up your talk and Eric's talk. So regarding your latent representations, if they can predict quite well, can we think from Eric's perspective about like small models to better interpret what's doing there? Is that possible? Yeah.
Oh, I see. So you're asking about how to interpret what's going on in those latent representations?
Yeah.
Yeah, so I think that is challenging. I think in practice, a lot of the interpretability, so you sort of have to leverage the model itself plus the latent representations if you want to get good interpretations, I think. The latent representations themselves can be used to train some downstream task, right?
Yeah.
But if you want to then connect back to the original data and ask questions like, what part of the high C matrix is it looking at?
Yeah.
That requires not just the latent, but all the weights in the encoder as well. And, usually some kind of backpropagation or at least ablation.
And I also wonder whether the foundation model can guide us to find the optimal resolution to measure. Because we know that if you want to measure a higher resolution, there's a higher cost, right? A higher sequencing depth associated with that. Can we find that guidance from the foundation model in some way?
I'm not sure because I think, what's optimal is not obvious. It often comes down to you have to have a very specific question.
Yeah.
And then the optimality might, I don't know if you even need the model to make that.
Or people can choose what technology to use for their experiment.
No, I don't think it can help with that.
Okay.
I don't know, not that I can think of.
Okay. Thank you. So